Polymers rarely act alone in a biological system. They assemble into many different structures. Take a protein, for example. It may bind stably to another protein, and only the resulting protein complex has biological activity. An example is hemoglobin, which is a tetramer of 2 alpha hemoglobin subunits, shown in blue, and 2 beta hemoglobin subunits, shown in purple, that together carry 4 iron groups that can bind oxygen. Defects in hemoglobin lead to anemia. Proteins can also bind sugars or polysaccharides, same thing, leading to a very important class of macromolecules called glycoproteins. Antibodies are glycoproteins. You can see the polysaccharide in the middle of the molecule. Glycosylation is a key variable that makes antibodies more or less potent as immunological drugs. Proteins that have enough non-soluble hydrophobic regions can also bind lipids, yielding lipoproteins. This is in fact the only way lipids can be transported through bodily fluids. HDL and LDL, high-density and low-density lipoproteins, for instance, transport cholesterol and are thus very important in cardiovascular health and diseases. Finally, proteins can also be complexed with RNA, yielding ribonucleoproteins. Because RNA can easily bind to specific DNA sequences, proteins that interact with DNA often have an RNA component. An example is telomerase, the enzyme which elongates the tips of the chromosomes called telomeres. This is an important function in avoiding cell aging and cell death observed in cancer cells, and is thus the focus of much research in oncology. A living cell contains thousands of different polymers in a very crowded environment. In fact, a cell can be considered as one single giant structure of interacting polymers, a very exciting and challenging entity, just like you and me.